Hello and welcome to Nikolai's genetics lessons, and this video is going to be about population genetics. And here is the problem. The mean internode length of the Abet binder variety of barley was found to be 3.2 millimeters. The mean length of the Asplant variety was 2.1 millimeter. Crossing two varieties produce F1 and F2 generation with average internode length 2.65 millimeters. About 6% of the F2 had internode length of 3.2 millimeters and another 6% had length of 2.1 millimeter. Determine the most probable number of gene pairs that is involved in the internode length, and the approximate contribution of each gene makes to the phenotype. If you think that you can solve this problem, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So first of all, how I'm going to show you how this problem is solved. I want to show you formula first. This is very easy. You can solve this problem under maybe 1 minute. And then I want to explain you the logic, because most likely you would forget the formula, because we tend to forget something that we don't use in our daily life. But logic you probably would remember much longer. So here is the formula. So extreme genotype and phenotype can be expressed as 1/2 raised n and squared. As you see in our problem, we have about 6% of F2 generation of one of the extreme phenotypes, that is 3.2 millimeters, and another 6% also we have extreme phenotype, that is 2.1 millimeters. So we have two extreme phenotypes, and both of them makes about 6% of the total. So we can use this number for our calculations. So here on the left we can put the 6%, or we can convert this number to the 0.06, that is the same. So here is frequency of the extreme phenotype, and here is a formula how we are going to find how many allelic pairs control this trait. Internode length. So in order to get rid of this square, let's take square root from both sides. So let's take square root here on the left side and square root here on the right side. So what we are going to get on the left side, so if we take a square root of 0.06, we are going to get 0.24. And on the right side we are going to get 1/2 raised n. Once again, n here stands for the number of allelic pairs that control this trait. We have to find n. So now we can find reciprocal of left side and right side of this number and this. So what is a reciprocal? Reciprocal is such a number when we multiply, for example, this number by its reciprocal, we are going to get 1. And when we multiply this number by its reciprocal, we are going to get 1. So this is very easy. We have to divide 1 by this number, and this is how we would find reciprocal. So if we divide 1 by 0.24, we are going to get a rounded number, that is 4, and reciprocal of 1/2 would be 2. If we multiply 1/2 by 2, we are going to get 1. So 4 equals to 2 raised n. So now you can easily see that n equals to 2. So if we square 2, we are going to get 4. So n equals to 2. So the trait under control of the two genes, and each gene has two alleles. For example, here we have gene A, and gene A has two variants of the alleles, dominant A and recessive allele A. And we also have gene B that also has two variants of this gene, dominant allele B and recessive allele B. So what we had at the beginning, let's analyze our problem once again. We had one variety that had 3.2 millimeters internode length, and another one had 2.1 millimeter internode length. So let's assume we now know that this trait under control of two allelic pairs or two genes, because this is diploid organism, each gene present in two copies. So what we can suggest that genotype of first variety that has internode length of 3.2 millimeters were capital A, and capital B, and capital B. As you see, here we have gene A and gene B. And when we cross this with a second variety, that is asplund, with internode length of 2.1 millimeter, we can suggest that genotype of this variety were small a, and small b, small b. Once again, here we have two genes, gene A and gene B, that is present here in homozygous recessive state, and second gene also present in homozygous recessive state. But total number of alleles is 4. So this genotype produce, as you see, 3.2 millimeters internode length. So that means that we have to divide 3.2 millimeters by 4 alleles that contribute to this trait, and we are going to find that each dominant allele contribute 0.8 millimeters. So in other words, we can say 0.8 plus 0.8 plus 0.8 plus 0.8, and we are going to get 3.2 millimeters internode length. As for the second variety, we know that asplund variety had 2.1 millimeters internode length. So once again, we have to divide 2.1 by 4 alleles. This time this is going to be 4 recessive alleles, and we also can find how much each recessive allele would contribute to this trait. And as you see, each recessive allele would contribute 0.525. So let me use rounded number, that is 0.5. So each allele here would contribute this number, and when we add all the 4 alleles, we are going to get 2.1. So as you see, now we can easily explain why in F1 generation, when we cross these two parents, we had this internode length, because genotype of the F1 generation were as follows. So let me clean the space here. So when we cross these two parents, F1 generation were capital A, small a, and capital B, small b. Because this is additive trait, once again, dominant allele contributed 0.8 and recessive allele contributed 0.525, 0.8 for the dominant allele B, and 0.525 for the recessive allele B. And when we add these four numbers, we are going to get this number 2.65 millimeters that we got in F1 generation. And also this were the mean of the F2 generation. And as you see, when we cross two extreme genotypes, this is parent 1 and this is parent 2, so this is parental generation. In F1 generation we are going to get only one type of genotype, so 100% of the F1 generation would have this genotype. But in F2 generation, when we cross this genotype with itself or self-pollinate, we would have many different genotypes, and we would have a distribution of those genotypes. For example, if the trait would be under control of one gene with two variants of alleles, and we had parent 1 capital A, parent 2 small a, F1 generation would be capital A, small a. So basically what we would have in F1 generation would be only this genotype that would be intermediate between parent 1 and parent 2. But if we self-pollinate F1 generation, we are going to get some distribution in the next F2 generation, and according to Punnett square, our genotypes would distribute as follows: capital A, capital A, capital A, small a, capital A, small a here, and small a here. So as you see, 25% would be one extreme genotype, 25% would be another extreme genotype, and also we would have intermediate genotype in the F2 generation if this trait would be under the control of only one gene with two variants of the alleles. So if additive trait would be, for example, dominant allele A would add 2 centimeters and recessive allele would add 1 centimeter, so the length of this plant would be 4 centimeters and length of this plant would be 2 centimeters. And in F1 generation we would have 2 plus 1. So this would be intermediate 3 centimeters, and if we self-pollinate we would have a distribution as follows. So 25% would be extremely large or 4 centimeters, 1/2 would be intermediate or 3 centimeters, and so this is 25%, and we can also say 1/4 or 25% also would be extremely small, so would be 2 centimeters. This would be distribution of the of this trait if we would have this trait under control of one gene with two variants of the alleles. But in our problem, as you see, we had two genes and in F1 generation we had genotype that is capital A small a and capital B small b. And these parents in this generation, we know that parent 1 and parent 2 will have the same genotype, because this is the only one possible genotype in F1 generation. So in order to find how many different genotypes is possible in F2 generation, we have to find how many different gametes this F1 generation can produce. And the first variant would be capital A and capital B. Second variant of the gamete would be capital A and small b. Third variant would be small a and capital B. And the fourth variant would be small a and small b. And of course, this is going to be parent 1. We have to cross with the same variants of the gametes of the parent 2 that can be produced by the same genotype, and we know that in F1 generation all the plants would have this genotype. So the second parent also may produce the same gametes, capital A, capital B, capital A, small b, small a, capital B, and small a, small b genotype. And now we can build a Punnett square. This time this is going to be 4 by 4. So square 4 by 4. And of course that means that here we would have 16 cells with different genotypes. And distribution would be more even. So if we build a distribution grid, the more genes we would have, the more even distribution would be, and our distribution would resemble bell-shaped distribution. And here we would have extreme genotypes, that is capital A, capital A, capital B, capital B. Another extreme genotype would be small a, and small b, small b. And all the rest, I don't want to take your time, but you understand that the rest would be different variants of the intermediate between these two extreme genotypes. And this extreme genotype, as you see, would occur in 1/16 frequency, as long as this one also would occur in 1/16th frequency, because here we have 16 cells, and each extremely large genotype would occur in this frequency, and extremely small genotype would also occur in this frequency. So now we can use our logic, and even without formula we can find how many genes, how many allelic pairs control the trait. As you see, when we had only one gene, extreme genotype equal to 25%. Here we have extreme genotypes that equal to about 6%. So quarter here and 1/16 here. So as you understand, with each new gene that controls the trait, with each new allelic pair, number of gametes, so here is the gametes, variance of the gametes, and here is the variance of the gametes, would double. So if we would, for example, see that trait is under control of 3 genes, and F1 generation would be capital A, capital B, small b, and capital C, small c, that means that here we would have double number of gametes. So instead of 4 we would have 8, and our square would be 8 by 8. So once again, extreme genotype would occur in the frequency of 1/64. So this number would quadruple, and the same rule applies with each new gene that controls the trait. So if we would have here capital D and small d, we would have instead of 8 by 8, we would have 16 by 16 square with different genotypes, and of course, once again, number of variations of the all possible intermediate genotypes would quadruple when number of gametes would double. And now I hope that even if you forget the formula, you still would be able to solve such problems using this simple logic. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.